pen is not working. What happened to this? फ्रान मेरा पेन देखना मेरा पेन नहीं काम कर रहा. Let me just इसको करें. Okay, done. Shall we start? Okay, 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 okay. One, first. Okay, so question number one is regarding it's related to transpiration basically, and this instrument is known as potometer. The instrument that we use for transpiration is potometer. Okay, so what is actually done in this one? The plant is kept in the water, and later on we'll check the speed of transpiration or the speed of water evaporation by the bubble. So we have to create a bubble over here, and when this bubble moves, so it would show the rate of transpiration. Okay, now if you look over here, they have mentioned here air. That is basically the air bubble, and the starting position is this one, and the ending position is this one. Okay, now I guess our first part is really okay. Complete the column heading in table 1.1. What should be coming over here? Position of meniscus. No, first of all, heading, start position, centimeters. Yes, centimeters. Okay. What is the starting position and what is the ending position? 1.4. Yes, it's a double one. Okay, and this one is 4. Okay, whenever there is a like a term 4, 5, 6, just add point 0 after this so that it would be more clear. Okay, in chemistry as well, you have to put that decimal. Okay, calculate the distance moved by. How much distance is covered? Like if it's moving from 1.4 to 4, what's the total distance covered? Just subtract it. That would be 2.5. No, 6. It's point 0.4. 2.6. And what would be unit? Centimeter. Okay. Now, how to calculate the rate of movement? How do we calculate rate? It's related to physics. Whatever distance over time. Okay. So the distance. What is the time given? Let's see. How much time is given? 20 minutes. Yes. So 2.6 divided by 20. Yeah. Rate is always determined by distance over time. Okay. Have you divided? Minutes is okay because they have given us minutes. And centimeter is also okay because we are taking the readings in centimeter. So it would be, I guess, 0.13 centimeter per minute. Keep in mind you have to mention the units. These are very, very important. Okay, the student had difficulty making an accurate reading of the position of meniscus. Now, what is meniscus? Meniscus is basically the upper layer. In the colorless substances, we actually take readings from the lower meniscus, and in colored substances, we take reading from the upper meniscus. Okay, and we should. How do we take reading? Our eye level should be uh, b- basically parallel to that meniscus. We should not be looking from top or bottom. It should be on the level. Okay. So state what this difficulty was and a way of preventing this difficulty when set up the apparatus. Now we are using water, right? So water is transparent. Exactly. Exactly. That would be more visible. Okay. Water is transparent. Moreover, what difficulty can be there? Because we are using a capillary tube over here. Here, the capillary tube is really small. Okay, the diameter, or we can also say the capillary tube is small. Capillary tube. There is single P in capillary. Is small. Okay. Okay. Now, how to overcome it? For the first one, we have to use colored liquid or colored water. We can use dye to make it colored. And when we use the capillary tube, because it is very small, we can use the magnification glass. Okay. Okay. We have to keep in mind that it should be on eye level, so there won't be any error. It's the first thing that we need to keep in mind while taking the reading. Okay. The stem was sealed in a tight fitting rubber bung so that its cut end was in the water as shown in the diagram. Describe what might happen if the seal was not leak proof. Now, it may happen that the rubber may have some leakage. So, what effect it would have on your reading? So, the effect is if it is not waterproof, it means water can leak out. Okay, and it can give us the wrong results, the anomalous results. Okay, what was I writing? Water could escape. Water can leak out. Okay, of the rubber band. Why haven't they used the word band? Okay, and its effect would be uh, wait. The effect on the plant. Water may escape. Uh, water may escape from. May not escape. No. So we will write. Wait. Let me rephrase it. I'm trying to say that if the water is also evaporating from the rubber bung, then then the transpiration rate won't be clear. How to put this in words? Okay. Um, not okay. Not all the water is evaporated from the plant, right? Let's say not all the water is evaporated from plant, and it can give us false result. Okay. The tap on the reservoir of the potometer can be opened so that more water can flow into the capillary tube, moving the meniscus back to the start of the scale. This is when we do when we want to repeat the experiment, so that it can go back to 1. Okay. We add water from here. Here is a tap. We just open it. More water is added, and water level will move backward. Okay. So when we are repeating it, where is it? So we just open the tap and they take it. Why we are repeating the reading? We have to use the same term in every paper to check the anomalous result, to check reliability, ability, to check anomalous result. I don't know why my writing is not good today. Okay, next one. The experiment in figure 1.1 is designed to investigate the uptake of water of a leafy stem. Sometimes this same measurement is described as the rate of water loss from the leafy stem. Explain why the uptake of water and the rate of water loss may be different. Okay, it can be used in other processes. Wait, if it is used in other processes, then the uptake must be equal to the water loss. Oh, okay. Got it. Got your point. Got it. Your point. Okay. We can also say, uh, ठीक है. This one is fine. This one is fine. Or there is one more explanation that when we are cutting the, like when we cut the stem, we cut it underwater, right? So so that the connection doesn't break of the xylem vessel. So there is already stored water in the plant. Exactly, exactly. That's why the rate of uptake might not be. But this is also correct. This is also correct. Okay. Next one. There is a table given. A student measured the result of transpiration on the plant growing outside in a field during morning. Mean light is available. 4, 6, 8, 10, 12. As the light increases or the temperature, we can say, increases, the rate of transpiration also increases. Now calculate this one on the graph. So while you draw this graph, keep in mind you have to mention x-axis. Exactly. What would be on x and y axis? Time. And we have to give the its time, hours, and here would be, is there any unit for rate of transpiration? Then we have to write this. We have to, exactly, we have to write this. If it's, yeah, we have to write it. Okay, if it's given in the table, then we have to write it. Okay, now I guess you can draw it by yourself. We cannot like make it over here. Okay, the next question. Use your graph, or definitely we are going to have a reading of this once we make a line. Shall we try? Let's say we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. So there are 50, let's say. And we have time till starting from 4 till, okay, 4, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 6. Where is it? Here? I hope. One, 2, 3, 4, 5, 6, 7, 8, 9, 10. Here. 8, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10. Okay, is it 10.0? Yeah, okay. 6.00, 800, 1000. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Here 12, and definitely 1 p.m. So I'm writing 13 over here. Okay, now let's have the other one. Let's say it's point one. Okay. Yes, we can. Yeah, we can. We can. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Here is 1. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Here is 2. And then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 3. Okay, 1, 2. Oh, we have to go till 10. Then I have to change it. It's 2.0. It's 4.0. Okay, 6.0. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 8.0. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, 10.0. Let's have a reading. The first reading is at 1.1. Let's say here in the middle somewhere here. Okay, keep in mind, put only one dot. Don't make it like this. Just put a dot, okay, while making a graph. Otherwise, your graph would be considered wrong. Okay, at 6, it's 2.4. Let's say if it's 2 here, it's 2 point. Really? Why? Exactly. I don't know why, why my graph is like this. Let me check. Let me check if I can add more picture. Let me check from the original paper. No, my original paper also has these lines. No, 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 this is the other paper that I've opened. I have to open paper one. Wait, because it's really tough for me to calculate. Okay, where's the paper? No, mine is same. I don't know why, but it's the same. Okay. Then at 8 it is 4.5 must be here. Here. 6. Have you made the graph? 10. Okay. One, 2, 3, 4 here. Let's say it's here. I don't think so you'll be able to see the like 12 it's 9. 9.9 here. Somewhere here. Now if we just join, okay, while joining these lines, try to make a single line. Don't cut the line, don't double the line. This is one very important observation in the papers, that students they make multiple lines just to make them dark. There's no need to make them dark. Draw a single unbroken line. Okay? No, we have to. We have to, yeah. Because we have to take reading at 11. Okay, 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 okay. Oh my god. It's here, here. Mine is going, I don't know where. Exactly. It's 8 point something. Okay, next one. Let's say 8.4. I'm not sure, but it's around 8.4 or 8.6. Let's write 8.4 to 8.6. Though we don't have to give like the range, I'm just giving a range for like whatever your reading is, write that one. Don't give the range in the answer. This would be considered incorrect. Okay. Leaves have many microscopic openings called as stomata, from where the water evaporates. Use this information. What happens? Okay. What is happening? Why the rate of transpiration is increasing? Light intensity increases, which causes. No, 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 no. We have to relate it with stomata because in the question they put focus on stomata. Then we have to explain it according, which causes the stomata to open. Actually, what happens, which is the actual process when the light falls on the leaf, it start photosynthesizing, which actually causes the ions to move into the stomata to open it. Okay, it's about the movement of ions, which you are going to. Exactly. Osmosis would occur and guard cells become turgid, the stomata would open. Okay. Light intensity cause the stomata to open. Your first point. Okay. With more light intensity mean more food is prepared, more stomata are open. Okay. That's why the rate increases. Okay. A student reads the rate of transpiration is lowest when the air around the leafy plant is still. Mean when there is no air, the transpiration is low. Investigate using the same apparatus to discover whether the statement is correct. What changes are you going to make? It's, I guess, 6 mark question. You have to plan an investigation. What are you going to use? What would be your dependent variable? What would be your constants? What would be your independent variables? Okay, 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 okay. So, what do? We use same, because we have to use the same apparatus. Leafy stem with, okay, with different wind intensity. And how? Okay, wind inten how we are controlling this wind intensity? How do we? By using, there's so many instruments. Fan, fan. Exactly, fan, hair dryer, using fan. We can also use it on cooler mode. We have advanced one. Okay. Okay. Fan and hair dryer. Okay. Then, what was I going to write? To check its effect on the rate of transpiration. Now we have mentioned both the independent and independent, dependent and independent variable. Okay. Okay. And we have mentioned that the conditions would be same, the same plant. Now, the temperature, what should be constant temperature? Light intensity, let's say. That is controlled by this tap and this one, the reservoir, this one. Okay, that is already controlled. Okay, what can be added? Temperature can be because earlier we discussed light intensity as well. Now we have to keep this thing constant. And humidity, the third factor which actually affects the um, rate of transpiration, should be kept constant. And the rate of transpiration was measured for the given time or for a specific time. Okay, measured for the same period of time for all conditions. Period of time for all conditions. Okay, now you will get your 4 mark. What two other marks should be added? Now you have to tell how you are going to check the reading. Same meniscus one. Take the initial reading, initial measurement, the initial, no, the initial meniscus was measured and compared with the final, right, with the final results. Okay, to check the rate of transpiration. Okay, and then we have to repeat to check the anomalous result or to check the accuracy or reliability. Okay, this is all that you have to write. Okay, identify the independent variable that we have already written. What is the independent variable? Wind. Wind speed. I have written here intensity. That would be the wrong word. Speed is the correct word. Okay, next one. A student wanted to discover whether some liquid collected from a vein in a green leaf contained glucose. What test we are going to use? And how do we perform it? Benedict solutions. Okay, solution. What do we do it? Is added to a sample. Yes, we have to give the whole, describe the test, whole process. Heat it, okay. Then what is the, because they have asked about the result, how it would show. The end color or the end result is brick red, brick red, okay, color. Or we can also write the brick red precipitates are produced. Okay, next one is you have to draw the diagram. There is no need of shading to make a shade to diagram. No shading is required. Okay, with the scale you can have the size of this. Okay, take the size of each and everything. Your size should be bit bigger than that. Okay, then keep in mind you have to draw these eyes. Okay, the tentacles, there are two eyes, two tentacles. It has to be okay. Okay, check the size. It should be like this. Okay, it should be like this here, starting from here till there, point B to point A. That is the important thing. point B to point A. You, because we are not giving the like scale over here, we just have to keep in mind that it should be like this. Okay, now the magnification. Now magnification depends upon this one, definitely, whatever your scale is. So it is 45 millimeter, let's say. It's 4.5 centimeter. So 4.5 centimeter is 45 millimeter. Okay. Then what we have to do on your drawing, draw a line, or then you have to draw whatever is your line. Okay, whatever is your size. Okay, use your measurement and calculate the magnification of this one. What is the actual size? Draw a line on the same location as the line B. Okay, the formula would be okay, and you have to find the answer. Done. So this is all for this paper. Let's move to the second paper, the variant 2. We'll discuss only the main experiment one in this one.